Ichthyophthirius multifiliis is the agent responsible for the disease known as ich, or white spot disease. This is a common disease found in aquariums. Ichthyophthirius is a genus within Holotrichia, which is a subgroup of Ciliophora. Members of Holotrichia are characterized by cilia covering the entire surface of the cell. It's a parasitic protozoan with a tropism for the epidermis and the lamellar epithelium. The infective stage of the parasite causes rupture of the epithelium. This can result in osmoregulatory disturbances and a decrease in gas exchange. In regards to epidemiology, the parasite has a multi-stage life cycle, which shifts between the environment and the host. All freshwater fish are susceptible to infection. Ancillary testing includes gill clips and skin scrapes. The image on the top right illustrates the life cycle of the parasite. The tomite is the environmental stage. It can be found adhered to the substrate, where it undergoes rapid division to produce large numbers of thyrans. Thyrans are the motile infective stage, which latch onto the gill epithelium or the epidermis and transform into trophons. The trophons are the feeding stage, which increase in size as they acquire nutrients. These are visible with the naked eye, appearing as white flecks on the fish. Once mature, the trophont will develop into a motile tomont, which will detach from the host and settle in the environment to become a tomite, thus completing the life cycle. The image on the bottom is a micrograph of a trophont. Notice the large horseshoe-shaped nucleus. This is a characteristic feature which makes it easy to identify. Clinical signs associated with infection include abnormal behavior and hyperventilation. Examples of abnormal behavior include hyperactivity, flashing, and rubbing. On gross examination, the epidermis and the gill filaments may be covered in white spots. Histologically, there are large numbers of intralesional trophons with a characteristic macronucleus. The infection is associated with lamellar epithelial hyperplasia, epidermal hyperplasia, epithelial degeneration, and pleocellular inflammation. The image on the right is an example of white spot disease in a neon tetra. Notice the white flecks on the dorsal and anal fin. This is a sagittal section of the head of a fish. You'll notice several parasitic protozoa that are embedded within the epidermis, along the dorsal aspect of the maxilla and the ventral aspect of the mandible. These are labeled in red. On closer examination, you'll notice several trophons. These are the feeding stages of the parasite. The trophons contain deeply basophilic cytoplasm and a large horseshoe-shaped macronucleus, which is characteristic of the parasite. Cryptocaryon irritans is the marine counterpart of white spot disease. The life cycle, pathogenesis, and appearance are all very similar. Notice the degenerative changes within the overlying epithelium, which is lifting off of the underlying dermis. Uronema marinum is one of the agents responsible for scuticulociliatosis, also known as uronemiasis. Uronema is another genus within Holotrichia of Ciliophora. This is a free-living protozoan. However, it can cause opportunistic infections during periods of immunosuppression. Higher rates of infection are associated with a variety of factors, including temperature fluctuations, transport, overcrowding, poor water quality, and concurrent infection. In regards to epidemiology, transmission is either direct or indirect. All species of fish within the marine environment are susceptible to infection. The parasite can be identified on a skin scrape. The image on the top right is a diagrammatic representation of Uronema marinum. The image on the bottom is an example of a skin infection in a tropical fish. If you look closely, you'll notice an area of ulceration in the dorsolateral aspect of the body. Clinical signs associated with uronemiasis include abnormal behavior and hyperventilation. Examples of abnormal behavior include flashing and rubbing. On gross examination, there are areas of color loss and excess mucus production. Other changes within the infected area include erythema and ulceration. Histologically, the most common lesion is ulcerative dermatitis. Large numbers of ciliates may be present within areas of inflammation. A characteristic feature of the parasite is deep tissue invasion. Where most protozoa remain near the surface, Uronema marinum can invade deep within the tissues. For this reason, the infection is often difficult to treat. Invasion is associated with edema, lymphohistiocytic inflammation, tissue necrosis, and hemorrhage. The animation on the right is an example of a fish with uronemiasis. You'll notice that the fish is hyperventilating. If you look closely, you'll also find an area of ulceration along the caudal aspect of the body. Within sections of the skin and subcutis, there are large numbers of ciliated protozoa invading deep within the tissues. Protozoa are labeled in red. Invasion is associated with hemorrhage, edema, and lymphohistiocytic inflammation. On closer examination, you'll notice the parallel rows of cilia that define members of the group Holotrichia. Here's an example of deep tissue invasion. You'll notice that the parasite has managed to cross the epidermis, the dermis, and the bones of the cranium, making its way into the brain. You'll notice that the inflammatory response is minimal. It's likely that the parasite was able to invade the tissues at such a rate that the body didn't have sufficient time to mount an immune response. Or perhaps the parasite was trying to escape the encroaching line of formalin fixation and continued to migrate through the tissues after the fish had already been euthanized. This could explain why the parasites are all lined up in a row. Trichodina is a genus of parasitic protozoa that belongs to the group Paratrichia of Ciliophora. Members of the group Paratrichia contain a ring of cilia around the oral cavity. Trichodina is responsible for the disease trichodiniasis. The parasite is a cause of opportunistic infections in immunosuppressed individuals. It has a tropism for the gill lamellae and the epidermis. The protozoa feed on mucus and detritus by scraping along the epithelial surface. This can cause irritation. In regards to epidemiology, these are commensal organisms on the gills and skin. Transmission is direct. All species of fish within the freshwater and the marine environment are susceptible to infection. The parasite can be identified on gill clips and skin scrapes. Clinical signs associated with infection include reduced growth and respiratory distress. Respiratory distress is more common with heavy parasite loads. On gross examination, you may see excess mucus production and thickened gill filaments. Histologically, there are large numbers of ciliated protozoa between gill lamellae. Gill infections are associated with mucocell hyperplasia, lamellar epithelial hyperplasia, and lamellar fusion. Skin infections may be associated with epidermal hyperplasia. With heavy parasite loads, affected fish can also develop ulcerative dermatitis. The animation on the right illustrates the feeding pattern of the parasite on the gill lamellae. Within sections of the gill, there are scattered ciliated protozoa between gill lamellae. These are labeled in red. On closer examination, the protozoa contain a skirt of cilia around the oral cavity. This is characteristic of Paratrichia. The epithelium adjacent to the protozoa is hypertrophied with mild hyperplasia. This is labeled in red. There's also telangiectasia of multiple gill lamellae. This is labeled in green. Paramoeba perurans is the agent responsible for amoebic gill disease. This parasite belongs to the group Amoebozoa. It's a free-living protozoan that causes opportunistic infections. Paramoeba has a tropism for the gill lamellae. Outbreaks are typically associated with elevated temperatures. In regards to epidemiology, transmission is either direct or indirect. This is a disease of farmed Atlantic salmon within the marine environment. Ancillary testing includes real-time PCR. The image on the bottom right is a transmission electron micrograph of an affected gill filament. Notice the extensive area of lamellar epithelial hyperplasia resulting in fusion of large numbers of adjacent lamellae. Clinical signs associated with infection include lethargy and hyperventilation. On gross examination, there's pale mottling of the gills. There's thickening of the gill filaments and excess mucus production. Histologically, there are large numbers of amoebae between gill lamellae. Amoebae are associated with extensive lamellar epithelial hyperplasia. A characteristic feature of this disease are what are known as kissing lesions. This is characterized by a symmetrical pattern of lamellar epithelial hyperplasia on adjacent filaments. Other lesions include lamellar fusion with pseudocyst formation, forming a pocket in which the parasite likes to reside. Within sections of the gill, there is marked lamellar epithelial hyperplasia and lamellar fusion, resulting in a significant loss of surface area. This would have had a significant impact on gas exchange. On closer examination, there is frequent lamellar fusion and pseudocyst formation, highlighted in red. Some of these pseudocysts contain parasitic protozoa. These are labeled in green. Closer still, there are large numbers of amoebae between gill filaments, which are admixed with mucus, erythrocytes, and cellular debris. These are labeled in red. Myxobolus cerebralis is the agent responsible for whirling disease, also known as blacktail. In Canada, Myxobolus cerebralis is a reportable disease. There are a number of watersheds in Alberta that are currently affected. Myxobolus is a genus of parasitic protozoa within the group Microsporidia of Myxozoa. The parasite has a tropism for cartilage. Infection is associated with granulomatous chondritis, which can result in skeletal deformities that cause compression of the nervous tissue. This is often associated with neurological disease. In regards to epidemiology, the parasite has a complex life cycle involving multiple host species. The ichthyospore is the infectious stage for fish. Whirling disease is a disease of freshwater salmonids, which serve as the intermediate host. Tubifex worms typically serve as the definitive host. Younger fish are particularly predisposed, given that their skeletons are largely unossified. Ancillary testing includes PCR. The image to the right is a simplified version of the Myxozoan life cycle. Myxozoans are actually related to jellyfish, both of which belong to the group Cnidaria. The Myxozoan is able to infect the host via highly modified nematocyst, which is the stinging organ of the jellyfish. The ichthyospore injects the sporoplasm into the fish, which undergoes development, eventually giving rise to myxospores via sporogony. During this time, the parasite causes extensive tissue injury. The myxospores are released into the environment upon the death of the fish, and then they go on to infect tubifex worms. Within the tubifex worm, the parasite undergoes sexual reproduction, eventually giving rise to ichthyospores. The ichthyospores then go on to infect other fish, thereby completing the life cycle. Clinical signs associated with whirling disease include abnormal behavior and reduced growth. Examples of abnormal behavior include whirling. On gross examination, characteristic lesions include a hyperpigmented tail and skeletal deformities. Histologically, there are large numbers of intralesional myxospores. These are associated with granulomatous chondritis and cartilage necrosis. The inflammatory process can also extend into the underlying nervous tissue, so associated lesions include meningoencephalomyelitis and neuronal degeneration. The images to the right are examples of some of the lesions you're likely to encounter, including skeletal deformities and hyperpigmentation of the tail. This is a sagittal section of the head of a fish. The labyrinthian system is highlighted in red. On closer examination, there is an extensive area of tissue necrosis and associated inflammation involving the skeletal elements surrounding the labyrinthian system. This is labeled in red. Closer still, there are large numbers of myxospores within the lacunae of necrotic cartilage. These are labeled in red. The infection is associated with granulomatous inflammation. A remnant of viable cartilage is visible at the bottom of the section. This is labeled in green. Destabilization of the skeletal elements would have likely impinged on the adjacent nervous tissues and sensory structures, resulting in disequilibrium. Parvicapsula pseudobranchiola is the agent responsible for parvicapsulosis. This is a myxosporian of the subphylum Myxozoa. It's a primary pathogen with a tropism for the pseudobranch. Outbreaks typically occur in the winter. In regards to epidemiology, the parasite has a complex life cycle, with fish serving as the intermediate host. The definitive host has yet to be identified, but polychaetes are a likely culprit. This is primarily a disease of salmonids within the marine environment. Ancillary testing includes real-time PCR and immunohistochemistry. The images to the right are sections of the pseudobranch containing myxospores within areas of inflammation. These are highlighted by the black arrows with the letters S and P. The smaller image in the bottom right corner is a section of the pseudobranch with immunohistochemical staining to highlight the pathogen in situ. The clinical signs associated with this disease include abnormal behavior, inappetence, reduced growth, lethargy, and blindness. Examples of abnormal behavior include swimming near the surface and disorientation. On gross examination, the pseudobranch is pale and swollen, with areas of hemorrhage and ulceration. If you look in the eyes, you might also see lens cataracts. Histologically, you'll find necrotizing and granulomatous pseudobranchitis, with large numbers of intralesional myxozoans. The image on the right is another section of the pseudobranch that's absolutely laden with this organism. Most of the epithelial cells have been lost. The blood supply to the eye passes through the pseudobranch, so a possible explanation for the cataracts is that massive infection in the pseudobranch causes disruption of the blood supply to the eye, resulting in tissue injury and blindness. Loma salmonae is the agent responsible for microsporidiosis. It's a parasitic microsporidian of the group Glugata of Microsporidia. It has a tropism for endothelial cells, particularly within the gills, though systemic infections are fairly common. Infection is associated with the formation of tissue cysts, known as xenomas. Rupture of the xenoma is associated with localized tissue injury and the release of microspores, which go on to infect other tissues. In regards to epidemiology, transmission is direct via ingestion. This is primarily a disease of Pacific salmon within the marine environment. Ancillary testing includes real-time PCR. The image on the top right is a diagrammatic representation of the microspore. One of the characteristic features of the parasite is the polar filament. This is the structure that the parasite uses to penetrate the host cell. Clinical signs associated with infection include abnormal behavior and respiratory distress. Examples of abnormal behavior include impaired swimming. On gross examination, you may find scattered white cysts in a variety of tissues. These are generally 0.5 millimeter in diameter. Histologically, lesions are more prevalent in the gill and may include granulomatous bronchitis, lamellar epithelial hyperplasia, lamellar fusion, and hemorrhage and thrombosis. The image to the right is an example of a ruptured xenoma. Notice the large defect in the tissue and the associated lymphohistiocytic infiltrates. If you look closely, all that's left of the xenoma is a single microspore. This is labeled in red. Within the section of the gill, there is a xenoma that's encysted within the main arteriole of the gill filament. This is labeled in red. Notice all the microspores present within. Within sections of the heart, there are scattered xenomas within the spongy ventricle. Some of the xenomas remain intact, with little evidence of inflammation. This is labeled in green. In other areas, there are discrete foci of inflammation and tissue necrosis, which are the result of xenoma rupture. This is labeled in red. Similar foci of xenoma rupture can be seen in a variety of tissues. Highlighted in red is an example of a ruptured xenoma within sections of the liver. Within sections of the brain, xenoma rupture is associated with an extensive area of lymphohistiocytic vasculitis and perivasculitis. This is labeled in red. Microsporidium cerebralis is another cause of microsporidiosis. As with Microsporidium salmonis, this is a parasitic microsporidian of the group Glugata of Microsporidia. This species has a tropism for neurons within the central nervous system. The disease is not well characterized, but in regards to epidemiology, transmission is believed to be direct via ingestion. This is a disease of Atlantic salmon in the marine environment. At present, ancillary testing is not available. Clinical signs associated with infection include abnormal behavior. Examples of abnormal behavior include slow spiral ascent and disequilibrium. There are no obvious abnormalities on gross examination. Histologically, there are large numbers of intralesional microsporidia within the central nervous system. The infection is associated with meningoencephalitis, neuronal degeneration, gliosis, satellitosis, and rarefaction of the neuropil. The image to the right is an example of a degenerating neuron with large numbers of intracytoplasmic microsporidians. Notice the large infiltrate of glial cells. Within sections of the brain, there are patches of meningitis. These are labeled in red. There are also patches of encephalitis, labeled in green. Affected areas are associated with congestion and a prominent infiltrate of glial cells and other inflammatory cells. On closer examination, there are large numbers of microspores with associated gliosis and rarefaction of the neuropil. Adjacent to the affected region, there's an area of intravascular thrombosis and fibrinous exudation. This is labeled in green. Nodular gill disease describes a condition that's associated with infection of any one of a number of representatives of the group Amoebozoa. The image on the right illustrates some of the many species of amoeba that have been identified as a cause of this disease. The amoebae are free-living protozoa that cause opportunistic infections with a tropism for the gill lamellae. Outbreaks are typically associated with elevated levels of environmental bacteria, so this is a disease of poor water quality. In regards to epidemiology, transmission is direct. Various species of freshwater fish are susceptible to infection, including rainbow trout and Arctic char. At present, ancillary testing is unavailable. Clinical signs associated with nodular gill disease include abnormal behavior and respiratory distress. Examples of abnormal behavior include swimming near the surface. On gross examination, the gills are pale and mottled. There's thickening of the gill filaments and excess mucus production. Histologically, there are large numbers of intralesional amoebae adhered to the lamellar surface. There could also be large numbers of bacteria within the interlamellar space. The infection is associated with filament bronchitis, mucocell hypertrophy, lamellar epithelial hyperplasia, and lamellar and filament fusion. Hyperplasia of the lamellar epithelium is associated with spongiosis and epithelial degeneration. Within sections of the gill, there's marked hyperplasia of the lamellar and opercular epithelium, with fusion of adjacent lamellae and adjacent filaments. This is labeled in red. On closer examination, there are large numbers of amoebae lining the surface of the gill epithelium. These are labeled in red. The infection is associated with hyperplasia of the lamellar epithelium, spongiosis, and vacuolar degeneration of individual epithelial cells. These are labeled in green. Tetracapsuloides bryosalmonae is the agent responsible for proliferative kidney disease. This is a parasitic myxozoan with a tropism for the kidney and spleen. Outbreaks typically occur with elevated water temperature. In regards to epidemiology, the parasite has a complex life cycle. Bryozoans typically serve as the definitive host. These are filter-feeding aquatic organisms, pictured in the bottom right. Freshwater salmonids typically serve as the intermediate host. At present, ancillary testing is unavailable. Clinical signs associated with infection include lethargy. On gross examination, the gills are often pale. This is the result of hypoplastic anemia. The kidney and spleen are enlarged and they may contain pale nodules. Histologically, there are large numbers of intralesional myxozoans which are associated with granulomatous inflammation. Within sections of the kidney, the hematopoietic tissue may be hyperplastic. In severe cases, there may be evidence of metastatic hematopoiesis. Within sections of the kidney, there's generalized histiocytic to granulomatous inflammation, causing disruption of the normal tissue architecture. Examples of this are labeled in red. Some of the renal tubular epithelial cells contain brightly eosinophilic droplets within their cytoplasm. This is an indicator of stress. On closer examination, there are large numbers of myxozoans scattered throughout the renal interstitium. These are labeled in red. Infection is associated with histiocytic to granulomatous inflammation and frequent leukolysis and degeneration of renal tubular epithelial cells. This is labeled in green. Kudoa thyrsites is a parasitic myxozoan that is responsible for the condition known as postmortem myoliquefaction. The parasite has a tropism for myocytes of the skeletal muscle. Although it has the potential to infect cardiac myocytes, this is fairly uncommon. In the live fish, Kudoa will produce large numbers of tissue cysts throughout the muscle. The cysts will rupture en masse within 24 hours after the death of the animal. At the time of rupture, the encysted myxospores will release a large amount of proteolytic enzymes. This causes liquefaction of the muscle, as illustrated in the images to the right. Unsurprisingly, there is little demand for such a product, resulting in a significant financial loss to the producer. In regards to epidemiology, the parasite has a complex life cycle. This is primarily a disease of salmonids within the marine environment. At present, ancillary testing is unavailable. There are no obvious clinical signs associated with infection. On gross examination, prior to the rupture of the cysts, you may notice large numbers of tiny white nodules. Once the cysts rupture, there will be extensive liquefaction of the skeletal muscle. Histologically, there are large numbers of encysted myxospores within skeletal muscle fibers. If the tissue cyst were to rupture prior to the death of the animal, this would be associated with granulomatous myositis and myocellular degeneration. Within sections of the skeletal muscle, there are small foci of myoliquefaction. These are labeled in red. There are scattered pseudocysts within skeletal muscle fibers which contain large numbers of myxospores. These are labeled in red. There's a discrete focus of histiocytic to granulomatous inflammation within the skeletal muscle associated with premature rupture of a tissue cyst. Free myxospores are occasionally seen in section. These are labeled in red. Pseudoloma neurophilia is another cause of microsporidiosis. This parasitic microsporidium has a tropism for the central nervous system and to a lesser degree the skeletal muscle. As with many other microsporidians, rupture of the xenoma is associated with localized tissue injury. In regards to epidemiology, transmission is direct via ingestion. Vertical transmission is also an issue. Susceptible species include zebrafish, which are freshwater fish that are commonly used in laboratory studies. At present, ancillary testing is unavailable. The image to the right is an example of a zebrafish with microsporidiosis. You'll notice the spinal deformity. This is the result of an infection of the spinal cord, resulting in neuronal degeneration and denervation atrophy of the skeletal muscle. Contortion of the body is the result of an asymmetrical loss of muscle function. Clinical signs associated with infection include reduced growth and emaciation. On gross examination, the most common lesion is a spinal deformity, including lordosis, kyphosis, and/or scoliosis. This is the result of denervation atrophy and chronic myositis. Histologically, there are large numbers of intralesional microsporidia. Associated lesions include meningoencephalitis, neuronal degeneration, gliosis, satellitosis, and rarefaction of the neuropil. This is a sagittal section of the brain. On low power, there's no evidence of disease. On closer examination, there is a xenoma within the white matter tract of the spinal column, just caudal to the brainstem. There is little, if any, evidence of tissue reaction, aside from mild displacement of a neighboring axon. This is labeled in red. Within sections of the brainstem, there is a single xenoma within the perikaryon of a single neuron. This is labeled in red. There's also spongiosis of the adjacent gray matter, which is labeled in green. Ichthyobodo necator is a parasitic protozoan that belongs to the group Prokinetoplastida of Euglenozoa. This is the agent responsible for ichthyobodosis. Ichthyobodo is an ectoparasite with a tropism for the lamellar epithelium and the epidermis. In regards to epidemiology, transmission is direct. The parasite has two stages, the free-swimming stage and the attached parasitic stage. This is primarily a disease of salmonids in the freshwater environment. The young and the immunosuppressed are particularly susceptible to infection. At present, ancillary testing is unavailable. Clinical signs associated with infection include abnormal behavior, lethargy, and inappetence. Examples of abnormal behavior include flashing and rubbing. On gross examination, there's patchy discoloration of the skin, which is associated with excess mucus production. Histologically, there are large numbers of intralesional flagellated protozoa anchored to the epithelium. This is associated with epidermal hyperplasia, lamellar epithelial hyperplasia, and mucocell hyperplasia. The image to the right is an illustration of what patchy discoloration might look like in an infected fish. This is a result of a thickening of the cuticle and epidermis in response to the parasitic infection. Within sections of the fin, there is patchy thickening of the epidermis. On closer examination, there are large numbers of flagellated protozoa anchored to the epidermis. These are labeled in red. The infection is associated with epidermal hyperplasia and degeneration of individual epithelial cells. These are labeled in green. Within sections of the gill, there is widespread lamellar epithelial hyperplasia with associated lamellar and filament fusion. This is highlighted in red. On closer examination, there are large numbers of flagellated protozoa adhered to the gill epithelium. These are labeled in red. The infection is associated with lamellar epithelial hyperplasia and degeneration of individual epithelial cells. This is labeled in green.